Hi, I am Jose Almanza from Harmony Schools, and this is my PBL project video about viruses, more specifically about the influenza virus. The purpose of this project was to have a greater understanding of the influenza virus. According to this project, in order to achieve that, I had to create a virus model. So I did. In order to create the virus model, I had to utilize materials that were given to me by the school. Even though the school was generous in both quality and quantity of the materials, I still had to acquire some of my own. The first thing I did was cut off 1/4 of the styrofoam ball in a 90-degree angle. This is about how much I removed. Here's a perspective shot. Then I colored the sphere everywhere but the inside. I used a marker, but I wouldn't recommend it. For less mess, I would recommend something more like clay or any other material. Next, I measured the shape of the opening on the inside of the sphere on both sides on colored and textured paper. Then I proceeded to cut them out. I then put ponding strips on the paper and then glued it on the sphere. I used map pins and beads to make the lipid envelope in the RNA. I inserted the pin into the bead-like cell, and then I attached it to its required place. This is how the model looked like when I was finished putting all the pins in the right places. Notice that I used certain colors for certain organelles, like yellow for RNA and various others for the lipid envelope. Here's a different perspective shot. I then attached pins with and without beads on the outside of the cell. The ones with beads, those represented the hemagglutinin, and the ones without them represented the neuraminidase. Here are some perspective shots. I then printed out the names and cut them out, and attached them with bonding strips to the model on the required places. That is how I created my virus model. This project has allowed me to gain knowledge about influenza viruses and thus answering a lot of questions about them. In my website, I have written down numerous questions and answers in a very detailed and easy to read way. I will include some of them here. For example, what are viruses? Viruses are an effective agent that typically consists of a nucleic acid molecule and a protein coat. It is too small to see by a light microscope, and it's able to multiply only within the living cell of a host. Its only job is to kill living organisms. How do viruses work? The virus injects its own RNA into the cell, causing the cell to do what the virus makes it do, which would make more viruses. When the cell makes more viruses, it dies. This sets off the body's immune system, which will try to get rid of the virus with fevers, diarrhea, and throw up. Other viruses attack different types of cells. For this project, I have kept a journal recording all the procedures, all the advancements, and even all the frustrations of making this PBL project. Again, all the information is uploaded on my website. The H1N1 influenza virus was the cause of the Spanish flu, and the Spanish flu was the worst medical holocaust ever recorded in history. It was even worse than the Black Plague. This pandemic killed as many as 25 million in its first 25 weeks. In contrast, HIV or AIDS killed 25 million in its first 25 years. I include more detailed information about this on my S.O.S connection in my PBL website. Thanks for watching Harmony Schools.